Something very special to be told about Debbie. She's going to talk about industrial biotechnology, about the opportunities there. But she is the initial recipient, the inaugural recipient of the Rosalind Franklin Award, now given at the Biotechnology Organization annual meeting for leadership in industrial biotechnology. And this is sponsored by the Rosalind Franklin Society. So for over the last 22 years, Debbie has made some incredible contributions to Novozyme, the company's fundamental knowledge, expertise in gene expression technology using bacterial and fungal systems. She's quite rightly the director of expression technology, genomics, and bioinformatics at Novozyme's research center that's located in Davis, California. She's got about 40 patents, and I think for the industrial side, that's equivalent to publications for granting tenure. So I'm hoping that the numbers of patents means that you'll get promoted if that's part of the career. She's published a lot of articles in the scientific literature as well. So Debbie, I'm looking forward to hearing from you.
I'm going to talk about something completely different, and I want to thank the organizers. This meeting has been, to me, extremely inspiring because you're going to hear what I do is completely different than what everyone else has talked about. But I hope to communicate to you how exciting and fun it is and how passionate it is to be in industry, and particularly using biotechnology to solve industrial problems, not to discover new drugs and to produce new drugs. So what I want to start out with first, I want to actually thank both Bio and the Rosalind Franklin Society for establishing this award. If you go to the industrial biotechnology meetings, you still see that the majority of speakers and conveners and panelists are men, often tall white men, no offense. And I think we really need to change this. And I think this is a step in the right direction to recognize women that have taken a leadership role in this industry and are moving it forward. I want to first start with a story about how I ended up in industrial biotechnology, because actually I've always been interested in science and math, even as a young child. And I used to get pulled out of class for special math and science advanced courses. And I like to say that the majority of students that were pulled out in my school were girls. They weren't the young men. So we had quite a strong group of young women that I went through grammar school and high school with. In high school, I had a tremendous physics teacher, Father Foley. I went to a Catholic school. I had 2 years of physics in high school, and that became my passion. I really wanted to be a physicist. I went off to UC Davis and started as a physics major, and after a year and a half, I realized I didn't feel like I fit in. I looked around. All of my classmates were young men, extremely bright, but maybe not the best from an emotional, intelligent point of view. And I had a real hard time seeing myself having those colleagues for the next 30 or 40 years, because I'm a very people person, a very team-oriented person, so I wanted to feel like I was part of a team. So I actually did some exploration into different sciences, and I found my home in what was called bacteriology then. They don't call it bacteriology. The departments are now called microbiology, and I thrived there, and I became very passionate about that. When I finished my undergraduate degree, actually, I again had a sort of a fork in the road, because I wasn't sure, did I want to go to graduate school so I could focus on research, or did I want to go to medical school? So I worked for 2 years at SRI International doing oil field microbiology. I had a great mentor there, Dr. Steve Rosenberg. I was fresh out of undergraduate, and he gave me so much independence. He actually treated me as an equal, asked my opinion, and it was a tremendous experience. Based on that experience and the fact that I knew I wanted to have children, I wanted to have a balanced life, I actually decided to go to graduate school to get my PhD in microbiology, and actually went back to UC Davis. I had a tremendous mentor there, Dr. David Agreziak. I studied actually a portion of the signal recognition particle, the RNA portion of that tells the cell this protein needs to be secreted outside the cell. When I was finishing my PhD, actually, at that point, my husband had just gotten a promotion. So I took a postdoc at UC Davis in Dr. Dan Kleonsky's lab, where I actually studied how proteins got localized to the vacuole. And it's a cousin in yeast to the lysosome that you heard about yesterday from Anna Maria, actually. And again, he was a tremendous mentor for me. And I was only in that postdoc for about a year when one of the women faculty members said to me, you know, there's this biotechnology company, it's a Danish company, but they're going to establish a research site right here in Davis, and they're looking for people with your expertise. And at that point, it had been drilled in my mind by my professors that the route to go was academics. But I thought, what the heck, I'll throw my name in the hat, go for an interview. It'd be a great experience for me. And I must say, I went for an interview. I had an offer 2 days later, and that was 22 years ago, and I haven't looked back, and I don't regret it for a single moment. It's been a wonderful, wonderful experience. And if it doesn't come through, I truly love my job. And I love the company that I work for. So what is industrial biotechnology? So we rethink everyday products every day. And what's shown in this picture, all of the colored items in this household and outside are things that industrial enzyme products have been used either to wash, to manufacture, produce, or to feed. You can see the chickens down there running around because animal feed is an area that industrial biotechnology touches. This should be updated actually, and there should be some crops or vegetables shown there, and those should be in color because now industrial biotechnology, whole microorganisms can be used in agriculture to improve yield. And we actually formed a bioag alliance with Monsanto this last year, Novozymes did, where the goal is to discover whole microorganisms that can be used in agriculture to get more from less, if you will. Okay, so I want to tell you a little bit about Novozymes. It's a global company, has more than 6,200 employees. We make over 700 products that are sold in 140 countries. It's a very R&D intensive company. We have about 7,000 patents, and 13 to 14% of our sales are reinvested back in R&D. It's a very profitable business, and we're actually majority owned by a Danish foundation, the Novo AS Foundation. We were founded in 1925, actually, and we were listed on the stock exchange as a separate company, Novozymes, in 2000. And what's shown down here is some of the main industries, but I'll get to that in a moment in more detail. Okay, in brief, our products are industrial enzymes or the whole microorganism. And when it's a whole microorganism, it is a wild type organism. It is not genetically modified. If we're selling the whole microorganism, that doesn't mean that it hasn't been mutagenized and crossed and various other things, which we could discuss. Probably means it is perhaps equivalent to a GMO, but it is considered a wild type organism. The majority of our business today is enzymes. It accounts for 90% of our turnover. We're the market leader in all the main industries with about 47% of the market share. And I've already talked about these things. We are a global company. We have production around the world, but our main sites are in USA, China, and Denmark for production. We support more than 30 industries. This just shows the different areas: household care, agriculture and feed, biopharma, leather, pulp and paper, textiles, wastewater solutions, bioenergy, and food and beverages. And more in detail, this just shows the different products and diversification within those areas. So in household care, not surprisingly, enzymes are used in laundry detergent. So 99% of the laundry detergents that are on the shelves either have our enzymes in them or our competitors and our enzymes combined. They're used as degreasers in larger industrial settings. The whole microorganisms are. In food and beverages, they're used in many different applications for baking, brewing, wine and juice, and dairy. Bioenergy has been a big growth area for us. So that's starch ethanol, making ethanol from corn. But a more recent area that we focused on, not that recent, it's been 10 years ago, is in biomass conversion. And actually, my area, the 4 departments that I direct, have been very much involved in this aspect of it. And then I just want to mention that, so these are our more traditional industries, if you will, but we're always looking for new industries that there's a synergy between our technology platform and those new industries. And a couple examples is the biomass conversion, where we can break down cellulosic biofuels, bioag, which I just talked about earlier, animal health and nutrition, and biopharma. And you could ask, what is the next big thing? We're always looking for that. As I mentioned, we are a global company. Our production is global, and we also are very global in our R&D setup. So the largest number of R&D employees are at our site in Denmark, in Bagsværd. Our second largest actually right now is where I am in Davis, California, but we also have R&D in North Carolina, and we've expanded there because of the BioAg Alliance with Monsanto, and we have R&D in Japan and China and India and Brazil. We're all over the map. So the wonderful thing about working for a global company is a lot of our projects are not only cross-functional, but cross countries. And that means that there's a lot of opportunity to collaborate. So we all heard yesterday, which I think actually cemented some of the thoughts in my mind, is women are oftentimes more communal, and this is a great industry or a great sort of system for women to function in, because we like to work together collaboratively in teams. And this is a wonderful opportunity to do that. So we don't just do innovation for innovation's sake. It is for the benefit of our customers. And our customers get many benefits from our products. It can be a cost reduction, is one possibility, to reduce their energy consumption, their water and chemical usage. It allows them to have some new products, new claims, things they couldn't produce before. And for many customers, something that's also become very important is the sustainability aspect of it. So by using the solutions that we and our competitors are putting forward, many companies now can truly claim that they are offering sustainable solutions. And this just actually puts a number on this. So today our products are helping to reduce CO2 in every industry actually that we are offering products to. And we actually have been, sustainability is the core, actually, one of our values. And so everything is measured. For every new product, there's a life cycle analysis done. Okay, but we don't do this in isolation. We know that the most successful products are products that we have worked together with our customers actually to develop. Because the customers, of course, know what it is that they need. They know what their problems are, what their aspirations are, the new products that they want. So many of our products we develop together with our customers. So our bioinnovation is inspired by nature. We find the microorganisms either out in nature, typically out in nature, or sometimes with non-cult organisms from the genomics, the genomic DNA sequence that's been done on non-cults. We grow them in big fermenters from one microorganism to trillions, and this is the top of a large fermenter. Our fermenters are typically about 160 cubic meters, so they're quite large vessels. And we either produce enzymes, microorganisms, or more recently, chemicals. And I'm going to tell you a little bit about the chemical area. Our technology platform really has 3 legs. It's a gene technology or expression, recombinant DNA, if you will. It's microbial techniques, and it's fermentation technology. So the ability to be able to take a microorganism and upscale it into that large of a production volume is really science and art. And we have a long history of doing that. We have a broad, actually, range of microorganisms that we use to express our products. Everything from bacterial strains, typically Bacillus, but sometimes E. coli. We have different fungal systems, Aspergillus, Trichoderma, Fusarium. Fusarium is an interesting one. It was actually developed at our site in Davis about 20 years ago. It is, the biomass of this organism is corn. I don't know if you all know what corn is. It's been eaten in the UK as a protein substitute like tofu for decades. It was approved about 15 years ago in the US for the same thing. So if you go into the frozen food section and you're looking for vegetarian, you'll see products that have corn in it. And it's actually the mycelia of this fungus, actually. And it's quite a productive host for enzyme production also. And we also use various different yeast. And the name of the game for us is that we want to take a particular enzyme and we want to produce as much of that enzyme as we possibly can from one of our large fermentation tanks. And we're constantly asked if they tell us, okay, we want you to make a strain that will produce 20 grams per liter of this product. That's going to make the product economical. As soon as we've gotten 20, it's guaranteed they're going to come back and say, well, can you double that? Because now we found other applications, right, that we want to use it in, and we need a higher dose. So it's very common that we are working constantly to try to improve yields and to improve the potential of our host to produce actually enzymes. And that involves everything from changing the different elements that are on the recombinant DNA that we put in. For instance, the promoter that tells the cell how much RNA should be transcribed before you make the protein. From translation, how do you go from the RNA to the protein the most efficient way? And then secretion, how do you get that protein outside of the cell? So over the years, I've worked on a number of projects, and a lot of the patents actually are related to techniques to do that, to improve yield. And I was quite interested in Jennifer's talk yesterday because, of course, this genome editing and being able to multiplex it is potentially something that could definitely be a game changer for industrial biotechnology. So I want to briefly talk about one of the projects that we've done recently, which is to produce malic acid using a filamentous fungi. So malic acid is currently made from a petro-based feedstock, and we would like to be able to take starch or biomass and convert that to glucose and then make malic acid. The market that we initially were going after was for food and beverages. There's a much larger market, which is for polymers you can make from malic acid, but the price of that would have to be, the production cost would have to be much cheaper. So the reason this was interesting to us is that out in the literature, actually there's a strain reported of Aspergillus oryzae. That's a fungus that we have a big toolkit to work with. That was a known malic acid producer. As I mentioned, we have an in-house toolkit extensively to engineer this host. There's also a genome sequence available. There was an Affymetrix chip, which allows you to look at the messenger RNA level of all the genes in the cell. And Aspergilli, actually, Aspergillus niger has been used for decades to produce citric acid. So it's known that it can produce a lot of acid. And it can utilize xylose, and it's a grass organism. So if you look at the metabolic pathway based on the genome sequence and what's known about metabolism, the most efficient way to make malic acid is to use a reductive TCA pathway that is in the cytosol versus the pathway that's in the mitochondria. So what we did was to look at what are the key components potentially that are required to get more glucose, if you will, going this way through the pathway leading to malate production versus going to the mitochondria. And so these are some of what's circled are some of the key components that we went after, including a transporter that would take the malate that's produced inside the cell and moving it outside. What happened actually was, it's not that easy that you can look and say, okay, I'm going to take these 3 genes, I'm going to hook them up this way, I'm going to put them in the cell, and I've solved everything. We actually did 75 different gene combinations, various insertions and deletions in the genome, and those were all screened to find the best malic acid producer. And then we used another very, very old tool in our kit, which is to do classical mutagenesis, to take the microorganism, to mutate it, and then again, using high throughput screening, we actually screened more than 50,000 mutants per week. We were able to find strains that were making even more malic acid. So I just want to show you the final result. So in the literature, there was work where folks were trying to produce malic acid in E. coli and Saccharomyces cerevisiae, and you can see the titers here, gram per liter, were 34 and 59. We actually ended up with the strain here. Here's the starting strain that we started with, which makes about, I think, 80 grams per liter of malic acid. And then by a couple of different recombinant systems where we overexpress different genes, and then by mutagenesis, we were able actually to get up to 350 grams per liter, which makes this economically viable. So we were extremely excited about this. It's not yet commercialized. We are hoping to commercialize it, to find a partner for it, but it truly showed us that if we had a toolkit for a microorganism that was a natural producer of something, that was the place to start to do engineering from. So I want to briefly end with 2 quick slides to give you my opinion. This is not Novozymes' opinion, but my opinion about the opportunities for women and UMR folks in industrial biotechnology. And then I want to end up with what I think are still the challenges. So as I mentioned earlier, the research in industry is done in teams. So it's very collaborative. If you love working together with other folks to solve problems, this is a great industry to be in. Over time, I have had, in the 22 years, I've been very fortunate enough to work on many different projects, many different products, using different skills and allowing me to continually develop. I've even been involved in some of the business development and going out and meeting with customers and partners. So there's a lot of ability to learn a lot of different new skills as you're delivering on projects and products. If you look, in the 22 years I've been in the industry, when I first started, when I would go to these meetings with customers or collaborators, I was oftentimes the only woman around the table. Now when I go, there's usually at least 1 or 2 others, which is great to see. So I've definitely seen an increase in the number of women and the number of women in leadership roles, in management, entry-level management and above. The other thing is that there is a lot of folks that want to help you and mentor. I've had 2 great mentors in our company. They're both men. One is Randy Burka, Dr. Randy Burka. The other is Einar Beck Jensen. One is more communal, but he's very effective. People love to work with him. The other's a little bit more, perhaps, competitive, but they have been great. They've been both a cheerleader for me, but also have helped me over time to develop. The important thing is you have to ask for it. It usually doesn't come on a platter that someone is going to offer you. Working for a Danish company, I can't say this is true in all of industry, but in a Danish company, work-life balance is recognized. And it's not just that people are aware of it, they walk the talk. That's certainly not true when you get up in certain leadership positions, but we have, for women of childbearing age, there's great benefits, great maternity leave, and so on and so forth. Challenges. Although we've come a long way, there's still a long way to go, and I think this was discussed yesterday also. There's still, if you look in the C-suites of companies, or you look even at meetings of speakers, it's still predominantly men. Even though in the life sciences, as we saw yesterday, and it's publicly available, about 50% of the degrees, advanced degrees in life sciences, are going to women. So we definitely need to do something about that. The other thing that can be hard for for any scientist, whether you're a woman or a man or a UMR, is that in industry, it is the business that drives the science. So there are times your science is going fantastic and you're so excited about it, results are going great, but because of a business reason, strategically, you may have to stop it and set it aside. And that can be a challenge for people that are just coming into industry. Because everything is done in teams, success is also celebrated in a team. And that's a great thing, but for women, which we typically don't like to brag and boast, that means you have to be willing to step out of yourself and let people know what you have contributed. Because when it comes time for promotions and recognition, it's not just about the team succeeding, but what have you as an individual done? What are you bringing? What value are you adding? And as I mentioned earlier, still at industrial relevant conferences, women are underrepresented. There's no doubt about it, as are UMRs. At the last meeting I was at, I don't think there might have been 1 or 2 UMRs presenting, but not very many. So there's still quite a long way to go. With that, I actually want to end up with a quote. All of the people that work with me know I'm very big on quotes, and actually I always have one posted outside of my office. And this one I think is very relevant, at least for my career. It's from Thomas Jefferson. It says, "I'm a true believer in luck, and I find the harder I work, the more I have of it." So certainly I haven't been handed anything. I've worked extremely hard, but there's also been a fair amount of luck involved in where I've ended up, the timing of things. So with that, I'll say thank you.
Perfect. Any questions?
Do you go to many academic conferences to look for new talent for node design?
Yes, all the time. And I co-convene sessions at some of those so that I can also be actively involved in sharing also what we're doing. So that also makes them want to come to Nova Science. So, yes.
Curious as to why there's so few women, because there's so many women in the field.
Right, yeah. It's a very interesting question. I wish I had the answer. I can definitely say, if you go into the lab, for instance in my area, more than half of the people in the lab doing the work are women. But as you go up in the management at different levels up, it gets smaller and smaller. And I don't have any good reason why. I can say that if you look at Novozymes and diversity, and you look it up on the web, our company now has some targets specifically for women, percentage of women that should be in new leadership roles, and the percentage of non-Danish people that should be in leadership roles. Because of course, our company, being a Danish company, has a lot of Danish leaders, right, over the years. And so they're really looking to make it more global. But I wish I had a good answer for you as to why. I don't.
Have they worked on it in any specific way?
Yeah, so they've had over the years, they've definitely had some forums, which I was actually reading something, maybe it was in the Life Science Foundation magazine, a book about a group of women scientists who used to get together. So we did have a forum of women leaders, to get together and discuss issues, challenges, share information, and we also have some particular leadership development that they're targeting women to try to get more women ready for the challenge and the opportunities. So.
Thank you very much.